welcome. Uh, thanks for coming. This is our last lunch and learn of the first semester. Do me a favor, guys. Take seats. The countertop, we grow bacteria on there, so you don't want that on your butt. So we have a really special guest, and I'm glad a lot of y'all came out today. For those of you that are watching this later, this is definitely going to be one that you'll want to see. So this is Farah. Farah graduated from the Academy of Biotech 2 years ago, and we were remote, and was one of the group that was our first to actually pass the base, and is certified as a biotechnician's assistant. Now at University of Maryland, Farah works with these other two wonderful individuals in what's called the Gemstone program, which is part of one of the honors colleges, the University of Maryland. And through that program, they do research. They have to submit and they'll tell you all about it, right? They had to get money. They had to submit their research plan. They had to do a literature review for a project. And what they're doing is really cool, especially given that they're only sophomores. And that's awesome. So there's a lot of things you can learn from them. Everything from how do you prepare for college, what do you do once you get there? If you go to Maryland and you're in the Gemstone, what's it like? All the way up through what is it like to do research? What is glioblastoma? So I'm going to let them take it from here, and I'll let them introduce themselves. I'm going to go and participate and observe myself. Thanks for coming.
Thank you for the introduction, Mr. Silber. So we're just going to go ahead and jump into the presentation if you want to go to the next slide.
Want to get a show of hands. How many of you are thinking about going into research or exploring research in the fields of science or medicine? Can we just see a show of hands? Okay, that's exciting. A lot of you are interested. I'm going to ask this question again at the end of the presentation, so we'll see if we were successful in inspiring you. So.
Okay, so this is our agenda, just everything we're planning on covering for today. If you guys have any questions throughout the presentation, just feel free to raise your hands and we'll be happy to answer. Okay, so I'm just going to go ahead and jump into what Gemstone is and what the Honors College at UMD is. So could I just get a show of hands? How many of you have already applied to UMD or are planning on applying? Okay, so a good amount of people. So once you apply to UMD, if you get accepted, your acceptance letter might include an invitation from the Honors College. And what that is a group of 8 different living learning communities within UMD. These are just 8 of them. So there's some with like cybersecurity, there's a life sciences one, but the one that we are in is called Gemstone. So what Gemstone is a multidisciplinary 4-year research program as opposed to a couple of the other honors colleges, which are 2 years. And it is student-led research. So our freshman year, the majority of it was dedicated to brainstorming research ideas, and then towards the middle of the year, we would have a vote on which research projects we would be most interested in exploring for the next 3 years. And then we eventually narrowed it down to around 10 different research projects with 6 to 12 members per research team. And then each research team is assigned a research mentor, which is an expert in the specific field of what your research project is on. Our research mentor is Dr. Huang from the bioengineering department. He's worked with UMD's medical school on glioblastoma, and he knows a lot about what we are planning on looking at. So about our team, we are all sophomores. There's 8 of us, but just the 3 of us today. We have different majors throughout each of us. So I'm a cell biology major, Neta's neurobiology and physiology, Dylan is bioengineering, and our goal is to explore combinatorial treatments to reduce cell migration, invasion, and proliferation of glioblastoma. So I'll kind of just go through these slides quickly for the sake of time, but the scientific method is a cyclic process as opposed to it being linear. So whenever we get answers to questions, we try and go back, ask more questions, and see how we can build on that information. And these are kind of the steps of the research process. Right now we are at the fundraising and literature review step. So we kind of looked at what research has been done in the past, and we kind of brainstormed where there are gaps where we could start another research project, and we just finished fundraising money to start our actual hands-on research.
Hello, everyone. My name is Dylan, and I'm going to talk a little bit about some of the concepts that you need to understand before we go into our project. So the first is glioblastoma multiforme, also known as GBM, as we'll indicate throughout this presentation. And what it is it's basically an aggressive malignant tumor, or in other words, cancer in the central nervous system. And what makes it especially dangerous is the survival rate. So as you can see, it's only about 7% survival rate with average survival of about 8 months. There are conventional treatments that are being used, such as chemotherapy, radiation therapy, and surgery. However, studies have shown that these treatments are not completely effective and often leads to recurrence of the cancer. Um, and one of the reasons for this is called the blood-brain barrier, which is specific to the brain itself. You can think of it as like a protective layer around the brain that prevents all foreign antigens, not just drugs, but bacteria, whatever else, from entering the brain. In addition, as I mentioned, there was a surgery option for the treatment. That always doesn't work simply because these tumors in the brain are really deep beyond the skin. So surgeons would prefer not to go that deep and cause fatal damage to that patient, and therefore it often leads to the cancer further spreading. So we're looking of ways to improve this. And one of the therapies that we're looking into is called photodynamic therapy. It's a fairly modern cancer treatment that targets a specific region of the brain in which the tumors are localized. So it's a very localized region-specific treatment. And the way that it works is you have these things called photosensitizers, which you can think of them as light-activated drugs. And what happens is you put them inside your body, they spread throughout your body, and then they localize at the tumor region that you want to target. And what you do is you shine this laser, and the drug is then activated. In other words, activates something called the reactive oxygen species, which is like the killer agent. And as soon as it's activated, it starts to kill the cancer cells. So that's the kind of the treatment and therapy that we're going to be working with. Another important concept is called RTK inhibition, which is, in other words, receptor tyrosine kinase, which you can think of them as these receptors on the cell surface when you have these ligands or things that attach to the receptor and alert a bunch of signals through proteins that ultimately leads to an action. So you can see here through this diagram, the little brownish thing is the receptor, and those are all signal proteins that once it binds the ligand, then these signal proteins are then activated, initiating a response. So the specific RTK that we're going to be looking at is called AXL. And what this does is it basically blocks apoptosis, also known as cell death, in cancer cells. So you can imagine that if it blocks this death, these cells are able to multiply and spread throughout the entire body. And that's what's happening in glioblastoma and most cancers. So in theory, if we were to block AXL, then the cell death would then be induced and more cells would die, thus killing the cancer. So one of the inhibitors that we're currently looking at that specifically does this is called bemcentinib, and it's currently in clinical trials right now. It's a fairly new drug, sorry about that. And like I mentioned before, it kills these cells and inhibits cell migration and further proliferation.
All right, so I'm just going to go a little bit into the background of the treatment challenges that a lot of researchers have faced when trying to develop treatments for glioblastoma. So you'll find that in any case when you want to research any solution to a problem, you have to do a lot of extensive research into why the problem exists, what affects the problem, why hasn't it been solved already, what challenges do you have to address when you're thinking about how to develop a solution. So in this case, with a brain cancer like glioblastoma, there are a lot of challenges, and that's why there's currently no comprehensive cure for this type of brain cancer. One of the challenges is tumor heterogeneity. So I'm sure you're all aware of the term of like heterogeneous, which just means different distinguishable elements. Brain cancer tumors, especially glioblastoma brain cancer tumors, in every case they have slight differences, like honestly on a case-by-case basis. So with each patient, when you look at the tumor, oftentimes they have different degrees of aggression, different degrees of infiltration, but with most brain cancer tumors, they're highly infiltrative and invasive, which means that they spread really deep into the brain. And as Dylan mentioned earlier, when you're trying to develop a treatment that can kill those brain cancer cells, sometimes those treatments can actually negatively affect or harm the nerve cells and the neurons surrounding the cancer tumor. And you don't want to kill the healthy cells, in addition to killing the cancer cells. So that's the idea of developing like a targeted treatment in cancer treatment development is you want to develop a treatment that can kill only the cancer cells, right? So you want it to target only those bad cancer cells rather than the harmful, rather than the healthy cells and molecules surrounding that cancer's environment. And that environment surrounding the cancer is known as, the tumor microenvironment. So another issue that Dylan also mentioned earlier was the blood-brain barrier. You can imagine the blood-brain barrier, it's a barrier in your brain, but it's basically this wall of extremely tightly packed brain cells and blood vessels that prevents, it's almost like your cell membrane. It's very semi-permeable. It prevents any large bacteria or chemotherapeutic drugs from, going deep into the brain or passing the barrier, but it'll allow like small molecules like oxygen molecules. So with the case of the blood-brain barrier, you'll notice that in areas of the brain where there's a brain cancer, the blood-brain barrier actually becomes even less permeable because the cancer cells can signal pathways that make it so that that barrier is even stronger and can prevent any sort of chemotherapeutic drug from getting to the cancer and killing those cells. And when we say chemotherapy, chemotherapy is essentially just sending, different medicines and drugs or chemical substances into the body through like an IV fluid, for instance, and those drugs are meant to kill the cancer cells in some way. Of course, there's a wide variety of them because they target different aspects of the cancers. Another issue that we are studying in terms of a challenge to developing a treatment is the immunosuppressive microenvironment. So as I mentioned earlier, a microenvironment is simply just the area surrounding a cancerous tumor. And so with the case of glioblastoma brain cancer, whenever you have some sort of harmful agent that infects the body, our body has an immune system, right? It initiates some sort of immune response, such as, macrophages or white blood cells, T cells, for example, that can engulf or absorb those harmful bacteria or substances. With the case of brain cancer tumors and a lot of cancer tumors, they can actually signal pathways that turn off your body's immune response, or at least prevent it from working against the cancer cells. So you can, honestly, cancer is a lot smarter than we think. That's what we found out over the past couple of decades. They can develop these extremely strong defense mechanisms, and as we develop new treatments, they can even evolve and develop even more defense mechanisms. So it's really important that we understand those challenges really, really well and those defense mechanisms really, really well before we start developing a way to fight them. Another factor is tissue hypoxia. Hypo means low, so hypoxia is just low levels of oxygen. You'll notice around a lot of cancerous tumors, especially with glioblastoma brain cancer, they'll have like a lack of oxygen. The reason why is because, as you know, like stem cells can differentiate into specialized tissue cells. With the cancer cells, there are like, for example, they have basically their own stem cells. Like with glioblastoma, there's glioma initiating cells, which are essentially stem cells that can differentiate into glioblastoma brain cancer cells, given the right environmental molecules. So. Now, to jump into our research specifically, we are, based on our background research and our literature reviews into the topic of brain cancer research and existing treatments, we've found that with glioblastoma and other brain cancers, a monotherapy, which is just a single treatment therapy, has been proven to be extremely ineffective, and that's demonstrated by low survival rates after treatment. The cancer recurrence, there's a high recurrence rate after a patient receives treatment, so that basically means once you undergo some combination of chemotherapy, radiotherapy, and surgical resection or removal of parts of the tumor can easily and quickly grow back, even after just a couple months from your initial treatment. So our question is, if monotherapies aren't that effective, well, how can we develop some sort of combination therapy that can address multiple challenges that we just discussed and effectively eliminate the tumor and prevent its reoccurrence. Now, you'll see 2 research questions on the board. A show of hands. If you were going to conduct your own study, right, and you have a research question, who would choose question 1? I'll give you a chance to read it. Okay. Even if you don't understand what it means, just take a guess. Who would choose question, well, let's start with who would choose question 1? Okay, no one. Who would choose question 2? Who has no idea what question they choose? Okay, I would say you're all right. First of all, it's important to do research before you come up with a research question. However, there's a lot of elements that we considered. We actually, in the beginning, we had the first research question. And now, after conducting a little more research, we now have developed the second research question. So in general, when you're developing a research question, there are different things to think about. Does anyone have any idea what are one of the factors you'd consider when developing your own research question? That's okay. I would say, maybe if I asked you, if I said my research question was, how can we cure, all the cancers that exist in the world, that wouldn't be a very good research question. So you kind of want some specificity in your question. You also want it to be somewhat measurable or testable, right? If you're saying, I'm going to answer this question, based on the question, you have to be able to develop some sort of method or experiment to use to test it and be able to answer it. Another thing you want to think about when doing research in general, not just when developing a question, is that it's somewhat timely. When I say timely, I mean it's somewhat novel, or it's rooted in some sort of new technology, or it's based on an existing technology, but it's exploring some sort of new aspect of that technology, right? You're building off of existing research to, basically produce,
Provides activities by Montgomery County Recreation. All MCPS students can receive a free scholarship by using the code MCPSKID2022 during registration. Activities run from 8:30 a.m. to 4 p.m. Visit activemontgomery.org.
Sorry.
No, you didn't do anything. That was I'm just gonna drop this.
All right, so, yeah, okay, so we can. No, okay, great. So basically, as I just discussed, so our research proposal is going to be developing a combination therapy for brain cancer that addresses more than one of the challenges that more than one of the treatment challenges that we discussed earlier. So one aspect of it is, as Dylan mentioned, inhibition of receptor tyrosine kinase protein known as AXL, which will basically prevent recurrence and metastasis of cells by not blocking that programmed cell death, the apoptosis of those cells. And then the second component of our combination therapy is photodynamic therapy, which targets the existing cancerous cells around the tumor. Okay, so let's just jump into our methodology. I mean, whenever you think about a research proposal, you ask yourself, well, how am I going to test this? How am I going to do this? Well, in our question, we said that we were going to test, migration of those cancerous cells, which is essentially the ability of cancer cells to travel through fibrous proteins, sort of like the highways of the cells, highways of cell, and reach other areas of the brain and establish new tumor sites. We also want to target cell proliferation, which is the ability of cancer cells to undergo processes like mitosis and cell division, and basically reproduce and develop more cancer cells. And so with each of those targets, there are different methods, different experiments that you're going to conduct to measure each of those target variables. But how do we start? We'll start testing on cancerous cells, right? And what that's called is in vitro experimentation. In vitro basically means testing on living cells outside of an organism's body. So often research will start just on these existing cell lines. Then once we conduct tests on brain cancer cell lines, If they prove to be favorable, if they prove to be somewhat successful, then we'll try it out on an in vivo experiment model. In vivo simply means testing in or on living things. Obviously, you wouldn't want to start with humans. You'd start more with like an animal model. So we would use a brain cancer mice model. And then after conducting like in vitro and in vivo experimentation, we'd most likely have to actually repeat and validate some of those experiments again before we publish any of our results. And of course, even though the methodology is, targeting specific aspects, like we'll target, okay, with this assay, we'll target like proliferation of the brain cancer cells, we still have to keep in mind our which is to eliminate the brain cancer tumor and prevent it from recurrence. And then, of course, the larger goal in mind, which is to increase survival of the brain cancer patients. Okay, so another aspect of our project is developing we're going to employ nanotechnology, and we're going to develop some sort of nanocarrier that's going to be able to carry, our inhibitor to the cancer site. Our inhibitor and our photosynthesizer to the cancer site. So nanotechnology is an extremely, extremely large field. And what you'll find is there's a lot of aspects of nanotechnology. And it's basically understanding like materials on this very small atomic and molecular scale. Nanocarriers specifically are these small like capsules that can transport different drugs and different materials, and it can take it directly to the cancer site or to the cancer cell, or you can use it to target other molecules and receptors and proteins that are in the microenvironment of the cancer that affect its ability to grow and to divide and to reproduce. And so in this case, what researchers have found with using nanocarriers to deliver like chemotherapeutic drugs is that they can prolong the circulation of those drugs, and they can improve the drug solubility and its ability to target those cancer cells.
Okay, so this is just kind of the big picture, a big conclusion of our research. So the reason why we chose brain cancer is that there are some of the sickest patients with the worst prognosis, which means they have the worst outcomes. They have the current surgical procedures aren't sufficient to be able to remove all the tumor, and there's a high likelihood that the tumor will come back. And we focused on non-invasive and minimally invasive therapies because it's better for patients, it's less invasive, there's less damage to surrounding cells, and it minimizes the risks associated with open surgery. So in the future, survival rates, we're anticipating survival rates to double, and the continuation of this doubling is very promising.
Any questions? Thank you for listening, by the way. Sorry. Oh, and one more thing. If you can go one slide back. Sorry. Go ahead and you guys, if you guys want to, we have an Instagram page where we post updates. If you guys want to follow us at Team MetaBio. But yeah, thank you very much for listening.
So if anyone has any questions about their project, their research, about Gemstone, about what their transition was like, what they did in high school, like there's everything's on the table.
Yeah, so we have different small molecule inhibitors in mind. So let's say the first one doesn't work, we could try a different small molecule inhibitor, and then if we find that the small molecule inhibitor's target isn't necessarily the best one, we could try exploring different targets.
Also, one thing I want to mention is even though something doesn't work, doesn't mean that it isn't important. So we might look for a way to build a project off of this failure. If you could, yeah. But yeah, that's an option.